Hello everyone, my name is Lykke, and the next 15 to 20 minutes we will be muscling in on the mechanisms underlying exercise's beneficial effects on health, and I will focus on skeletal muscle glucose uptake. But before we get into this, I just want to take you back in time. So imagine that the year is 1960. You will not have been born by then, but let's imagine that your mom develops type 2 diabetes. You get money to spend on research. Would you spend this on A, developing an affordable treatment for diabetes, or would you spend them on conducting basic research on how bacteria protect themselves? I think that those of you who have watched the video that I sent up would probably all of you would spend the money on conducting basic research on bacteria. And the reason for this is that basic research can yield surprising results. And in this case, it provided us with the opportunity to produce insulin in a safe way. And I think that similarly, if we do the basic research on exercise and how exercise can, for example, stimulate glucose uptake, we might be able to harness the beneficial effects of exercise in many different diseases. So the aims of my talk are the following. I want to outline the molecular mechanisms by which exercise mediates glucose uptake. I also want to identify challenges for future basic exercise research that I think is needed for us to drive this further and to translate it into clinically beneficial new treatments. So who am I? My name is Lykke Sylow. I am Associate Professor at the Department of Nutrition, Exercise and Sports at the University of Copenhagen here in Denmark. I have trained with some of the best, I think, in this field, Erik Richter, Mary Klipp, and Bruce Spiegelman. I am on the editorial board of Journal of Physiology, and I'm also on the International Research Group on Biochemistry of Exercise, and we're organizing the IBEC conferences, and the next one is in Toronto, hopefully, next year. I'm also a mother of 2, and that means that my workstation often looks like this. And I'm also used to be a competitive beach volleyball player. I'm still active in playing, but when I was competitive, this was actually what spiked my interest in exercise biology and exercise physiology, because I wanted to understand what modulations occurred within muscle that I could use to improve my performance. But then I got an injury and I had to refrain from exercising for a long period of time. And I realized that by not exercising, I felt terrible, both mentally and physically. And I started to look into and be more interested in the health benefits of exercise. And I was overwhelmed when I looked at the literature about how many different conditions that regular exercise can actually prevent. So this shows how much reduced risk there is of all of these diseases if you exercise train. So while I was overwhelmed by this amount of evidence, I was also underwhelmed by the lack of evidence that there is on the molecular mechanisms that controls these beneficial effects of exercise. So there's virtually nothing out there, and it's really been difficult to dig out any results that proves how exercise prevent diseases. So I think this is an interesting topic to explore, and I think in particular the topic of how exercise increases glucose uptake in skeletal muscle is interesting because skeletal muscle insulin resistance is a major site is a major site that is impaired in diabetes, and also in conditions such as cardiovascular disease or type 2 diabetes, as we have talked of, but also several cancers are associated with marked insulin resistance, and sarcopenia and cancer cachexia as well. So this skeletal muscle insulin resistance is quite detrimental to these diseases. And one of my early aha moments in science many years ago was that insulin and exercise, the two most potent mediators of glucose uptake, actually increases glucose uptake via distinct mechanisms. And this was an aha moment for me, because that meant that in insulin-resistant muscle that cannot take up glucose, exercise could still increase glucose into the muscle, and thus could be a very potent therapeutic intervention. Yep. So some of the mechanisms by which exercise modulates health has been identified, although the mechanisms are really still lacking. And if you look at this, I want to divide the effects of exercise into acute exercise effects and chronic exercise effects. And if we first look at these here acute exercise effects, these comprise the effects of exercise that occur during the exercise bout, and I want to talk a lot about glucose uptake today. There's also the phase after an acute exercise bout that I'm sure that Rasmus Købstad is going to talk about in his webcast, that increases insulin sensitivity, and that can be for up to 2 days. Then there are the chronic exercise effects, which consist of increased capillarization and increased protein expression of many proteins that produces a more metabolically efficient muscle that occurs in response to chronic long-term exercise training. And what I just want to point out is that even if you do chronic if you're an exercise trained individual, you are an exercise trained individual because you're undertaking regular exercise. And that means within a given week, you will probably have undertaken exercise maybe one or two or three times. And that means that you will spend a considerable amount of time in this acute phase window, as illustrated here in these tabs, which in itself is also extremely beneficial for health. So I just want to point out that the health effects of exercise training is not only the long-term adaptations, but it also because you just spend a considerable amount of time in response to exercise or the acute effect of exercise. So the first evidence that exercise and insulin stimulates glucose uptake via distinct mechanisms came from when the insulin receptor was cloned and it was possible to knock it out in mice. So the Mirco mice have an insulin receptor knockout. So the insulin receptor here is removed genetically. And as you can see here, if you measure glucose uptake in response to insulin, control mice have a substantial increase in insulin-stimulated glucose uptake, whereas this is diminished in the insulin receptor knockout mice. And that does not go for exercise-stimulated glucose uptake, really suggesting that insulin is not needed for exercise to mediate glucose uptake. Further evidence for this increase in glucose uptake during exercise occurring independent of insulin resistance came from early evidence here in the type 2 diabetic subjects that were compared to normal, because in this study, and this has been shown several times afterwards, they saw in normal glycemic subjects that exercise significantly increased the translocation of the glucose transporter GLUT4 to the plasma membrane, which is a necessary step for glucose uptake. And that also happened in the type 2 diabetics. So this really proves that even in severe insulin-resistant muscle of type 2 diabetics, exercise is a potent means to increase glucose uptake. And finally, I just want to mention here, or not finally, but finally before I get to the real molecular part, I just want to mention that GLUT4 is a common distal event that needs to happen in order for insulin or contraction to induce glucose uptake. And that was evidenced after the GLUT4 was discovered and cloned in the 80s. Then GLUT4 knockout mice were created. And as you can see here, the normal insulin response on glucose uptake was completely abrogated in these muscles GLUT4 knockout mice. And that was the same for contraction. So even though insulin and contraction seem to elicit distinct upstream mechanisms to induce glucose uptake, the distal steps, such as GLUT4 translocation, is common between insulin and contraction. And that gave us the clue that it had to be some regulator of GLUT4 that was common to insulin as well as exercise. And here is where this rho GTPase RAC1 comes in. And I'll get back to this and talk much more about RAC1. Yep. And now I hope that I've convinced you that the insulin and exercise stimulate glucose uptake pathways, approximately at least, are distinct. And now I want to deep further into outlining the molecular mechanisms by which exercise mediates glucose uptake. And this is probably also a good time to grab a coffee and maybe stand up and sit down again, because it's gonna get molecular. Good. Great. So this is an overview of the molecular events that regulate glucose uptake in skeletal muscle. Or if I should correct myself, this is probably maybe 1,000th or 100th of what is occurring in response to exercise that might participate in glucose uptake, because we know very, very little about what is actually regulating exercise-induced glucose uptake. But what we know is happening is that that in response to an action potential, so this is neuron, and then here's an action potential that excites the myofiber, and when that happens, calcium is released from the sarcoplasmatic reticulum, which causes cross-bridge formation. And this cross-bridge formation elicits at least two important events for glucose uptake. One is that it produces a stress signal that occurs because the cross-bridge formation costs ATP, and that signal activates the AMP-sensitive AMP kinase. AMP kinase has for decades been believed to be implicated in exercise-stimulated glucose uptake because of its role in translocation of GLUT4. However, recent evidence suggests that it's probably not necessary in order for exercise to normally regulate GLUT4 translocation. So I'm not going to go much more into this pathway. Another pathway that we believe are increasingly and very, very, very important here is the mechanical stress signal that is also occur in response to cross-bridge formation because of the shear stress that contraction elicits on the plasma membrane. And this results in a mechanical stress signal that is sensed by integrins, and it is sensed by other complexes here at the plasma membrane that can then activate the rho GTPase RAC1. And RAC1, we know from some of the seminal studies by Amir Klipp's group in response to insulin, are a key regulator of insulin-stimulated GLUT4 translocation. I'm here to tell you that it's also very important for exercise-stimulated glucose uptake. But before I get into the evidence for this, I just want to mention also that RAC1 regulates a very important complex at the plasma membrane called NOX2. And NOX2 is important for generation of reactive oxygen species in response to exercise. And recently we found out that this complex might also play a key role in exercise-induced glucose uptake, and I'll also show you the recent data we've got on this. Yeah, so when we had this hypothesis that RAC1 would be involved in exercise-stimulated glucose uptake, firstly, of course, it was important for us to show that RAC1 was actually activated during exercise in skeletal muscle. And in order to determine this, we used a proxy for exercise-stimulated glucose uptake called PAC1. And as you can see here, PAC1 is phosphorylated, and it is phosphorylated at this activation site when RAC1 is bound to it and active. So this is a good proxy for RAC1 activation. And as you can see here, both in the soleus and gastrocnemius muscle, there was an increase in PAC phosphorylation in response to exercise in humans. So this was encouraging, and we then went on to generate RAC1 muscle-specific inducible knockout mice. So these mice lacks RAC1, but all other molecular signaling is fine. And in these mice, there was a partial reduction in exercise-stimulated glucose uptake. So that suggests to us that RAC1 is involved in exercise-stimulated glucose uptake, and now we wanted to understand the mechanisms by which RAC1 was regulating this process. So I'm just going to go back to this molecular figure, and now I want to zoom in here on the NOX2 complex. So the NOX2 complex is localized at the plasma membrane. It consists of several different subunits, and in order for all of these subunits to assemble and function, active RAC1 is required. And Thomas Jensen's group have been interested in ROS production during exercise, and last year they published the first study showing that in response to an acute exercise bout in humans, there was a substantial increase in the production of reactive oxygen species. So next we asked whether this induction of ROS depended on RAC1. So we went back to mice because this is the model that we can use to obtain mechanistic knowledge. And Carlos, the then PhD in Thomas' group, now a postdoc, he did treadmill running of mice and he measured ROS production. And what he found was that also in mice, there was an increase in ROS production in response to exercise. But that when you knock out RAC1, this was completely abrogated. So mice that did not express RAC1 could not at all increase their production of reactive oxygen species in response to exercise. And this seemed to be highly relevant for glucose uptake because mice that had a knockout of the P47 subunit of the NOX2 complex, which renders it dysfunctional, also had reductions in exercise-stimulated glucose uptake, similarly to the RAC1 knockout mice. Good, so we do believe that RAC1 is really important for exercise-induced glucose uptake. So this is a complicated figure and I've simplified it a little bit over here to the left. So we know that there is an important mechanical stress signal that we're only just beginning to recognize as a contributor to exercise-stimulated glucose uptake. And this activates RAC1 via unknown mechanisms, but we think it has to do with integrin signaling. And that then assembles the NOX2 complex to produce reactive oxygen species, which is necessary in order for exercise to translocate GLUT4 to the plasma membrane and for glucose to enter the cell. All right, so this is what we know now. And I think just drawing these really tiny lines here with these very few molecules has taken many, many years to identify. And I think we're only at the top of the iceberg. And the reason that I think that, and others I think that, is that if we do an acute exercise bout, which was done by David James' group, as I show you here, the amount of phosphorylations, which are often what regulates enzyme activity, that increases during just one bout of exercise are enormous. So every little dot on this figure is a phosphorylation site that is acutely regulated by exercise. And the interesting thing is that with these data, the known phosphorylation site only was 10% of what was identified here. So that means that 90% of what is going on in response to exercise, an acute bout of exercise, we don't know. And we don't know how it's regulated, and we don't know the outcome of this regulation. Similarly, with exercise training in mice that have undergone exercise training, which I just want to note is a very different thing from an acute bout of exercise. But here we also see all of these different proteins that are regulated, and many of them we don't know their function of, and we don't know how they could benefit health in response to exercise. So this gets me to my next aim, which is to identify some of the challenges for future basic exercise research. And one of the challenges that we have is that we're looking at a system with high degree of biological robustness. And this high degree of biological robustness occurs because of high degree of redundancy. And redundancy within a system is a characteristic of any complex biological system. And it's also often a characteristic of a system that is very important because it has to work. It's like biology's contingency plan. So glucose uptake in response to exercise is extremely important from an evolutionary point of view because it is what provides us with energy to move. And therefore, of course, if you remove one protein and then everything falls apart, you cannot take up glucose, that would render us extremely biologically vulnerable. So of course, there is a lot of biological robustness in this system, but it also makes it extremely difficult to identify therapeutic targets because it's difficult for us just to understand the basic biology of this system. Because if we knock out one protein, we might not see a phenotype because there are redundancy or other proteins that can take over. There are feedback mechanisms that can compensate. So this makes it extremely challenging to study, but I think still it is very, very important to elucidate the basic biology of the system in order to understand how we can harness the beneficial effects of exercise pharmacologically. All right, so just in summary, we have outlined the molecular mechanisms by which exercise mediates glucose uptake, and I've shown you that exercise-stimulated glucose uptake partially relies on RAG1. We've also identified some challenges for future basic exercise research, and I think that a major challenge is the identification of pathways and therapeutic with therapeutic potential in this complex system. That's going to be really difficult for future research. Okay, so finally, let's pretend the year is now 2020. You have all the money in the world to spend on research. Would you spend it on A, developing an affordable treatment for diabetes? Or B, would you spend it on conducting basic research on how exercise promotes health? I hope that my talk have inspired you to believe that if we can understand some of the molecular mechanisms by which exercise promotes health, for example, by increasing glucose uptake in an insulin-independent way, that we might become one step closer to harnessing these beneficial effects of exercise pharmacologically. And that will be have tremendous impact for society because we know that exercise can really benefit so many different diseases. So with that, I'm done, and I want to thank you for your attention. And I'm very much looking forward to meet you all in virtual reality soon. And here are just some questions that I put up for discussion. Thank you.